Welcome to Library Seminars, a platform for the presentation of research and ideas in support of the National Oceanic and Atmospheric Administration's mission. My name is Lisa, and I'm your NOAA Central Library host. Today's library seminar is part of the 2021 Knauss Fellows Lunch and Learn series, a monthly webinar where Knauss Fellows showcase their own research. Andrew Villeneuve, a 2021 Knauss Fellow working in NOAA Fisheries Office of the Assistant Administrator as a Fisheries Science Interagency Arctic Policy Fellow, will be our moderator today, and he will introduce our two speakers, Derek Kraft and Hally Stone, each of whom will speak for 25 minutes, followed by 5 minutes of questions and answers. Before I turn this webinar over to our speakers, here are a few logistical tips to help you enjoy our presentation. If you're having trouble with the audio or visual components of GoToWebinar, please log out and rejoin us. This will usually reset the software and resolves most technical issues. This presentation is being recorded and will be available on the NOAA Central Library YouTube channel by later today. We highly encourage you to ask questions, so the speakers will address them at the end of their presentations, so type them throughout the seminar. The questions chat box is located in the GoToWebinar control panel. So that last detail, I'll turn the presentation over to Drew.
Thank you for that introduction. So today I am very pleased to introduce our first speaker, Dr. Derek Kraft, who is a Knauss Fellow, and his position is within NMFS as a highly migratory species management division Knauss Fellow. First thing you know about Derek is that he likes sharks. He received his bachelor's degree in biology from Oregon State University and his Ph.D. at the University of Hawai'i, where he was stationed at the Hawaii Institute of Marine Biology. His research focused on conservation genetics of sharks and shark bite forensics. His talk today is entitled "When Sharks Bite, They Leave Evidence Behind: Forensics for Surfers and Shark Depredation." Derek, the floor is yours.
Hey, thanks, Drew. Welcome, everybody. Thanks for sharing your lunch hour with us today. So, as I get some technical difficulties, there we go. Yeah, so here's a little intro into my talk. It's a bit of a two-part talk. I'm going to mostly be talking about how shark bite forensics can help out in the surfing community and other recreational communities as far as when sharks bite people's surfboards and things. And then at the very end, I'll talk a little bit about some shark depredation work we've done over in the Marianas Islands. So as Drew mentioned, I was lucky enough to do my PhD out at the Hawaii Institute of Marine Biology. Now this island is pretty much the closest thing to Steve Zissou's research island in the film Life Aquatic. We have some shark pens over here, as well as there's a shark lab attached to those pens. I worked at a genetics lab up on top of the hill over here, and I worked with a professor out there named Carl Meyer. Now, coconut's really great, or sorry, HIMB is really great. We actually have to take a boat to work, and on those little boat rides, you essentially have a few minutes with whoever's on the boat with you that day. You're all sitting knee to knee, and on one of these boat rides one day, Carl says, "Hey, Derek, we are sticking surfboards in sharks' mouths a lot to, do bite impressions and study how that works. But do you think that there might be some DNA left on there, and we could possibly, acquire that DNA and identify the species based on that?" So I thought about that. I said, well, sure, we can try. I mean, if you're out there doing it anyways, we can try to collect some, and it could give some really important information. So a lot of There's some shark bites that happen around Hawaii. From 1995 to 2019, there's been about 120. Unfortunately, 6 of those were fatal. About 29 were either just a surfboard or a sup or a kayak was bit. And with all of those bites, we know that about 63 were tiger sharks. We had about 15 reef shark bites. We've had 2 cookie cutter shark bites. So with only 2 cookie cutter shark bites ever have all happened in Hawaii. If you don't know what that is, definitely look it up. But about 40 were unknown. So a third of the species from all of these bites, we don't know what kind of shark was involved. Now, if we zoom out a little bit and look at the global scene for that, from 2010 to 2019, about 70% of shark bites, the species is still unknown. And that's a huge data deficiency there. So we don't even know what species are involved. So if I'm a good detective and I'm walking onto a scene and I'm looking for evidence, what I'm really hoping is that there is DNA left behind. Because if there is, we can hopefully use it to identify what species was involved. So those are just kind of our main two questions. Is there DNA on it? And can we use it? So we went out, or when we think about a shark's mouth and the mucus in there, we generally tend to think that it's pretty sticky. And this has to do with how sharks breathe. So they're always pushing water in their mouth and out their gills. And while if their saliva wasn't sticky, it would essentially be washing out the whole time and they'd have to keep regenerating it. So it's nice and tactile so that it stays in their mouth. So we went out to test this theory. Here's intern Kaylee collecting some DNA for us. And if we watch this in slow motion, you can see the transfer of DNA happening. Okay, so then we bring that board back onto the boat and we use some sterile swabs. For any of you lucky enough to have had a COVID test, you've experienced these swabs up close and personal, but we rub said swab all over the board and hopefully try to collect that mucus. We store it in some extraction buffer, put it into the cooler, and take it back to the lab for processing, where we go through a process called DNA barcoding. So for those of you who don't know what that means, essentially what we can do is take some unknown tissue, or in this case, mucus. We can extract the DNA from those cells, amplify a specific region. So for this study, we were studying the CO1 gene, send it off to the sequencer. We get that sequence back and we can upload onto these huge data banks that are online. And that will match that DNA with known DNA sequences on there and give us the species of shark. So for our example there, we did it with that one surfboard, and of course we found tiger shark. So this is what results look like on that website. You can see the common name down there on the bottom and the scientific name on the left, and we can see that we got 100% match on a ton of hits. We are very, very confident that a tiger shark bit that board. So we set off on, so we thought we definitely need to test this a little bit more. Can we do this with multiple species? And can we also do it with more than just surfboards? And maybe things like wetsuits or other materials. So we set off on our 2020 field season, collected a couple of samples, and then we all know how 2020 went. Field season got shut down, but we happened to get 2 Galapagos sharks and that 1 tiger shark. Now, luckily, I had given the talk that I just gave to you guys to a bunch of peers at the American Elasmobranch Society conference. I gave it like in a hotel room over drinks one night. It wasn't even my main talk, but people really enjoyed it. And a young scientist named Lauren Meyer, not related to Carl Meyer, but same last name, said, "Oh, I work down in Australia and we have sharks biting stuff all the time. Maybe I can help you guys collect some more samples." And I was really eager about that. So I sent her over a big box of samples, and they sure did. So here's them collecting some samples on some neoprene. I was told these videos might be glitchy, but that's a white shark chomping on neoprene. So they worked in New Caledonia, North Folk Island, the Neptune Islands, and they were really awesome at upping our number of species and our samples. So we were able to get a bunch of bull shark samples, some dusky shark, some tiger shark, and a lot of white shark samples down from the Neptune Islands. So they pull those neoprene, or they also did some surfboards for us as well. They'd swab it. They'd use 3 swabs for each bite just to make sure at least one of them was picking up some mucus somewhere in those bite marks. And they sent it back to us in Hawaii, where we processed them all. I had a lot of help from Marianne Webb. She's our lab tech extraordinaire. And for those of you who are into genetics, this is an extraction gel. So this little piece of information lets us know how much DNA is actually on those samples. And all those really dark lines are telling us we actually got a lot of DNA off of those swabs. And for those of you who've never seen what actual sequencing looks like, this is what it comes back as. This is called a cladogram. And all of those multicolored, all those different peaks, each different color represents a different nucleotide. So this runs through the machine. We look at all the different colored lines, and that's how we get our sequences back. That's just for fun. But here's our results. So we had 32 bytes total. And of those, 31 of them produced a successful sequence, like you saw on the last screen. We actually got a useful sequence from those. From 18 of the bytes, all 3 swabs worked, meaning all 3 samples from each byte was successful. From 9 of them, only 2 swabs worked. And for 4 samples, only 1 single swab was able to collect mucus. So it's good that we did all 3. From 1 sample from a white shark on neoprene, we actually couldn't get any DNA, no matter how many weeks I spent trying to get all of them to work. It didn't, so I accepted 96%, thought, okay, that'll be good enough. And you might notice this says successful DNA sequencing. This doesn't say successful species identification, but let me tell you why. I was running a Galapagos shark sample, and I expected to show up, oh, look at Galapagos shark, but I got some really interesting results. It said dusky shark, but also Galapagos shark, but also dusky, also Galapagos, 100% match. And I'm like, oh, okay, what did I do? Did I do the wrong one? Did I get contamination? No, none of that makes sense. Like, these are all nice, clean sequences. What's going on? And I did a little digging, and turns out historically, these 2 species have actually hybridized and intermixed, meaning their genes have intermixed, and their mitochondrial CO1 gene is actually identical. And not only have they done that in the past, they are continuing to intermix. So a cool study from 2019 found that these 2 species are still hybridizing. This is really rare in the shark world. There are like 6, there might be more, but that I've seen species that actually hybridize. So like the fact that we actually captured 2 of them was pretty amazing. You can tell these guys apart if you use nuclear DNA or something. We didn't go that far, but we said, hey, look, does this method work? You know, if we swab the bite, do we get the DNA? Yes, it does. So in conclusion for that study, is there DNA on it? Definitely. And can we use it to see who bit it? In most cases, as long as you can tell them apart at that gene. So that kind of shows this could be a fairly robust tool for these real life situations. And by robust, I want to say, you know, we were on the boat, we would collect the sample, it'd go right into the cooler, you know, and then into the freezer. And that might not really happen in a real world situation. You might not have a cooler around or whatnot. So Lauren, when she sent the samples from Australia, luckily they got lost for like 5 weeks. So these samples with, you know, cooler bag and ice packs in there floated around Australia for like 5 weeks where it's nice and warm, and then finally ended up in Hawaii. And I thought, oh man, like, you know, they might all be ruined. But even after 5 weeks in a box without cooling stuff, they all worked. I mean, they almost all worked. So that wasn't an issue. That kind of reinforced, okay, cool, robust tool. So we continue to be human shark interaction ready. So we put together these forensic kits and handed them out to local authorities, hoping we don't need them. Now, I kind of put all of this together for my dissertation, and I was finishing up at HIMB. I had taken my last boat ride. I had given my advisor a hug. I defended December 4th, and unfortunately, on December 8th, there was actually a fatal shark bite over on Maui. A surfer was bit on the leg, and it also bit his board. And we started getting a lot of questions like, hey, are you gonna test this? So, you know, it took us a couple days to actually scramble, but some local authorities were able to go and actually swab the surfboard. So the surfboard went straight to the police station, where it sat in some air conditioning for about 49 hours before they were able to actually go and swab that board. And they sent it over. So I stopped packing up my apartment, went back to the lab and ran these samples. And one of them, luckily, actually was able to collect some DNA. So from that fatality, we were actually able to confirm that this was a tiger shark. 100% no doubt, we know that. And that's the first time any of this kind of technology has been used in Hawaii, and let alone with an unfortunate event like this where there's a fatality. So we were able to supply that family and that community with as much information as we could about that incident. There's been a few more bites since then. This 2021's been off to a little bit of a bitey start. February 6th, there was a bite on Kauai. Just the surfboard was bit. We were able to get samples from that, as well as from a kayak that was bit about a mile offshore on Maui, a father and his teenage son. It actually sank the boat, and those two swam in from a mile away, which is terrifying for them. Local authorities were able to recover the boat, actually, and were able to swab that as well. And the description from the father of that event did not sound like a tiger shark. And upon further investigation, we measured the bite marks, and we actually have a lot of reason to believe that this might have been a white shark. So though Hawaii there's a lot of tiger sharks around, we occasionally do get white sharks. There's only ever been one other bite in Hawaii that was thought to be white shark, but that wasn't confirmed. So we have swabs from these. Unfortunately, they're not quite ran yet, but we have a team over there working on it. Marianne, our lab tech's doing a great job, and Madison Hemp is helping her with these. So those samples are being processed. I hope to have those answers pretty soon. And then comes the question everybody always asks, okay, well, do you think you could swab a human wound? And though we think we could, we are nowhere near ready to do that yet. There's a lot of issues around, things like patient care. Like if a person is bit, I am certainly not going to walk and be like, hey, can I swab you while you're getting taken care of? No, I'm not going to do that at all. There's definitely a lot of legal work as far as human test subjects that we'd have to get around. And not to mention how much paperwork goes involved to dealing with human tissue in our fish lab. We wouldn't really be able to process it, but we do think that this would work. And here's why. We've actually gotten it to work on fish. So out at HIMB, we have those shark pens. We have some captive sharks. We're able to kind of test this theory. So we threw some mullet in there on a string. This is a scalloped hammerhead, taking a nice bite out of it. We were able to swab the actual fish tissue and sort out the shark and fish DNA and identify, yeah, okay, that was a scalloped hammerhead that bit this fish. Now, though that kind of helped show that this would work for possible human wounds, it turns out that fish actually get bit by sharks way more than humans do. So here's my segue into the second part of the talk. Western Pacific Regional Fisheries Management Council, so they're a fisheries commission in Hawaii, came to us and said, hey, over in the Marianas Islands, our fishermen are complaining about a lot of shark depredation. So what shark depredation is when a shark takes a bite out of the fish like you see in these photos. And it's a big problem for them. And they said, do you guys think you could help us out? So Carl came to me and said, well, could we apply this to that issue and maybe gain some more information? And shark depredation is a pretty complicated issue. There's a lot of facets to it. So we thought the best approach to this would be, first thing we need to do is just characterize it. From talking with fishermen down there, we didn't get a great idea of what species were involved. So if we don't even know who the players are, we can't really put all the puzzle pieces together. So we want to gain some more information, and from there we can go and hopefully find some solutions for these fishermen and other fishermen as well. We kind of thought of a two-pronged approach. We thought it'd be awesome if we get some film of this depredation happening. We can see the species. We can maybe see some behaviors that are happening under the water that we might have missed from seeing above. Also, this depredation could be happening kind of deep, so you never even get to see it. So we thought that that'd be really helpful. And of course, hand out some DNA swab kits. Now, the plan was for me to be there for several months throughout all of 2020, but that didn't work. So we're really happy that we put together all of these DNA swab kits. So the same thing we did with the surfboards, but we bulked them up a bit. I had a group of people help me put together 30 of these kits that were fully loaded for fishermen to put on their boats. And when they get this depredation, they can take a sample, store it in the cooler, and we can analyze it. So we went down, we did 2 fisherman workshops. We tagged along with the Westpac Council. They had 2 meetings already set up, so we just tagged along and said, hey, we're also here to talk about shark depredation. Does anybody want to come to a workshop? And we got 20 fishermen to sign up across 4 islands, so Guam, Saipan, Rota, and Tinian. They were really happy we were there. At first, they were really suspicious of us, but we got to know them a little bit. Said, hey, we think we're here to help. We have this solution. So I hitchhiked out on a couple fishermen's boats. I got out on 2 trips. The weather wasn't great. We were there for 3 weeks. And of course, this was at the beginning of 2020. We went in February, and after that, we couldn't go back anymore. So we're really grateful that we got these kits out to the fishermen. The fishing wasn't always great, and it was pretty choppy. I know this video is choppy, so I don't know if you're getting the full effect, but on a 20-foot boat, that was pretty rough seas. I was able to use the cameras one day, and here we see a mahi-mahi chasing a lure. This camera is actually on the fishing line, and stage right, here comes a gray reef shark. And that's it, unfortunately. I'd hoped to get a bunch more, but that's what we got. So because we weren't able to go back, the fishermen kept up and did a good job. This is James, he's a fisherman in Guam. So he's collecting a sample for us here. Even though he's still busy fishing, he takes the time, swabs the fish. I think James was also our only fisherman to fill out his data sheet. I was really thankful, so thank you, James. Okay, so we got about 29 bite samples back from Guam and Saipan, and we're in the process of running them now. Marianne's hard at work with Madison by our side. So far, we've extracted the DNA, and we've found a lot of the samples had good DNA on there, but we haven't got the shark sequences back yet. I was hoping they'd be ready by this talk, but they weren't quite. And that's where that story ends for now. We hope that this is a good demonstration that this tool could be useful for shark depredation, and that when you get the fishermen on board, they can be real allies throughout the process. I have a ton of people who helped pitch in with these projects. Sea Grant gave us a lot of funding, Save Our Seas Foundation, Westpac Council, obviously the local authorities in Guam themselves, both in Guam and Saipan, they were really, really helpful. My HIMB community, both the labs, and with that, I have plenty of time for questions.
Thank you, Derek. That was really interesting. Well, I have certainly some questions. But before we get there, audience, we will take the next, let's say, 7 minutes to answer your questions. So please type them in the questions chat box, and I will read them to Derek. Before I do that, I also want to encourage you to download Derek's slides, which are located under the handouts drop-down menu in the control panel. So let's see, I did think we got some questions. Let me look at that right quickly. Okay, so the first question asks, do you worry that this type of identification could lead to conservation implications? For example, an increase in targeted fishing / attacks on tiger sharks if they're confirmed to be behind a large portion of attacks?
That's a great question. And that was one of my first questions when I was introduced to this project. I was like, well, wait a minute, what are we doing here? Are we playing the blame game? And I think the answer is no, in my mind. So if the answer to reducing shark bites was cull sharks, which some places still believe that to be the answer, it would have worked. We would have done that already, and it hasn't really made a difference. I think the goal, especially, the goal with shark bites on surfers and that type of stuff, that's mostly to just complete the story, right? That can be pretty important to the family and to the community. And as far as the shark depredation, that's to give us more information as far as like, okay, let's say we find that it's silky sharks doing a lot of the shark depredation. that allows us to go in and do some more research. You know, we can specifically target silky sharks and tag them. Maybe find some solutions as far as, hey, they're not around this time of year or this time of day, or they're not around this area, and give the fishermen a little bit more information on maybe where they can go or what times of day. So though I hear your point, I don't see it as a big issue, and I do think that this research can actually provide a lot of good information.
Thank you. And the next question asks, did the cookie cutter sharks bite people or surfboards? They are usually found in deeper waters.
Yes, great question. The cookie cutter sharks have bitten people twice, and it was both times people were swimming the channel between Molokai and Oahu at night. And so they do come up at night a little bit, apparently, into shallower water. So yeah, they did bite people, and it did end their swims, unfortunately.
Next question, have you tried your primers with human DNA?
Great question. I meant to make a buffer slide with this. Kind of like I said, I can't actually try them with human DNA in my lab, but what I did do is I aligned the primers with human DNA to look to see, hey, if these line up really good, they're going to amplify in the PCR. But when I lined them up, both the forward primers that we used lined up terribly. So I'm not worried about them amplifying human DNA. And actually, our paper on this is in the final review process with the journal. So hopefully it'll come out soon. And there's a figure in there actually showing that. So keep an eye out for that.
Awesome. Next question. What can the fishermen do to decrease depredation once they know the predator species? Thank you, and very interesting.
Yeah, so what the fishermen can actually do, I don't have an answer to. Like I said, that study was mostly just to try to paint the whole picture, and from there we can come up with solutions. We have mulled over some ideas. I mean, there's a lot. There are some research suggesting that electromagnetic currents can deter sharks a little bit. So for those of you that don't know, sharks have an extra organ that can actually sense electromagnetic currents. And there's a lot of advertising and things out there sold that are shark deterrents that, okay, it creates electromagnetic field and it turns the shark away at the last minute. They kind of work. They seem to work a little bit. I wouldn't put them on and go swim in sharky water, you know, because you're safe now. I don't recommend that at all. However, these fields could be set up, you know, they could be set up around the boat. They could be miniaturized into gear to go on the fishing lines themselves, but that's the holy grail. That's the idea for down the line. So, but the different species have different responses to different frequencies, and so if you know the species, you can better customize those sorts of tools. Yeah, but for now, we're still working on those solutions.
This next question asks, what new research questions do you have, or what's next in your research?
Awesome. Man, we would love to be able to test human wounds. We have started to look into the mounds of paperwork and regulations to do that. But that's kind of all I can see in the future for that so far. We might also continue doing this work around the Hawaiian Islands. So Carl's working on some funding to do that with local fishermen in Hawaii and anywhere else that's interested.
All right, we actually have two more questions. I'm going to read one of them, and then that will be our last question for this part of the webinar. The question asks, I know in recent years, eDNA has become a tool. Can your sample be contaminated if surrounding seawater gets on the sample or impact your findings? Good
question, very good question. And we've thought about this as well. We were worried, okay, there's a shark in the water, we stick the tip in the water, and now it's contaminated with shark DNA. We did some control swabs throughout this process. So Lauren, when they were down in Australia, if they had a wetsuit in the water and a shark came around, but didn't actually bite the neoprene or the board, they would take some swabs of the neoprene itself. And we never got any or enough shark DNA to amplify. We didn't see any on the gels, nothing amplified in the PCR. So we're pretty confident that when we do get DNA off of these swabs, that it was from the bite itself. I don't see it as a high enough concentration. Generally, when you do eDNA, you take 3 liters of water or a liter of water, and you filter it through a filter. So all of the eDNA gets on this little filter, so it's really, really concentrated, and that's how you pick it up. With just the amount of volume from a swab, I just don't think it's concentrated enough to amplify.
Excellent. Well, Derek, thank you so much for the answer to that last question in your presentation. I'm going to go ahead and transfer the screen to our second speaker now, and Drew will introduce her.
Great. Yeah, I just want to thank Derek again for that really interesting talk. That was certainly just really interesting to learn all about. Next up, we have Dr. Hally Stone, who is a Knauss Legislative Fellow in the Office of Senator Brian Schatz of Hawaii. Hally earned her Ph.D. in Physical Oceanography from the University of Washington in 2020. Her research focused on the broad impacts of coastal upwelling dynamics in the Pacific Northwest, with specific interest in how wind patterns affect continental shelf bottom water and phytoplankton biomass, as well as how harmful algal blooms are transported from known hotspots to coastal beaches. She hails from Buffalo, New York, and earned her bachelor's degree in astronomy and physics in marine science from Boston University in 2011. Her talk is entitled Linking Chlorophyll Concentration and Wind Patterns Using Satellite Data in the Central and Northern California Current System. Hally, I'll turn it over to you.
Hi, thank you for the introduction, and thanks to the organizers for the opportunity to speak at today's NOAA Lunch and Learn. And yeah, as Andrew said, I'm Hally Stone, and I'm in the office of Senator Brian Schatz from Hawaii. Today, I'm going to be talking about one of the chapters of my dissertation. So before I jump in, I wanted to, I wanted to just kind of highlight my co-authors in this, and this is a paper that is in Frontiers in Marine Science, if you're interested to read more about it. So Neil Banis of the University of Strathclyde and Parker McCready of the University of Washington were my PhD advisors. And then Rafe Cadilla of UC Santa Cruz helped with a lot of the biology, and Bridget Oval, who was our undergrad at UW, and she's now in grad school at Rutgers, helped with a little bit of the analysis. So in Seattle, where I did my PhD, fisheries are a huge part of both the economic and cultural identity of the city and state. The fish that you're most likely to see on a menu or flying through the air at Pike Place Market is salmon. But other fisheries like oysters, Dungeness crab, razor clams, and rockfish are important as well. And our link to the ocean through these organisms extends beyond economics, as historically the local fisheries were and continue to be integral to tribal communities. Washington State is part of a larger region that also relies heavily on fisheries. In 2016, the commercial and recreational fisheries supported about 269,000 jobs and contributed $34.5 billion to the economies of Washington, Oregon, and California. And all of these jobs and financial contributions are supported by the fisheries within the California current system. These fisheries are fueled by the highly productive phytoplankton that are found along the coast. And phytoplankton are the tiny algae that make up the base of the food web. So this satellite image of the Washington coast in Vancouver Island, BC, depicts a typical phytoplankton bloom from July 2014. And so you can see the phytoplankton is this green swirl that's very concentrated at the coast and kind of goes offshore by the Strait of Juan de Fuca. And for reference, this star marks the UW School of Oceanography, where I sat for 7 years thinking about fisheries from a physical oceanography perspective. And while this bloom extends a bit offshore near the Strait of Juan de Fuca, it is largely concentrated at the coast. And so why is that? Well, like plants that grow on land, phytoplankton needs sunlight and nutrients to grow. Unlike on land, however, where the plants can get their nutrients from the soil, in the ocean, these nutrients tend to fall down into the water column to depths without any light. And the continual struggle in the ocean is to bring these nutrients back up into the light so that the phytoplankton can use them. And so what brings the nutrients back? Washington's coastal ocean is part of the California Current System, or CCS, as I'll say probably throughout the rest of the talk. And it's one of the world's eastern boundary current systems. And the high productivity within the CCS is supported by coastal upwelling. And this is a process through which nutrient-rich subsurface water is transported to the surface by the wind. And so when the wind blows southward over the ocean, as it's showing in this figure on the left, it induces an Ekman transport offshore, and in turn induces a transport of this deep nutrient-rich water up to the surface, as you can see here in this figure on the right. And this nutrient-rich water supports the phytoplankton growth, with the wind's strength dictating how much of this nutrient-rich water is upwelled to the surface. And so more simply, when the wind blows equatorward over the ocean surface, it pushes the surface water away from the coast, which is shown here in tan. And then this deeper water is pulled up to the surface to replace it. And this deep water, now at the surface, carries those nutrients that are represented by the pink dots. And then so by pushing the surface water away from the coast, the wind is able to deliver nutrients up to the surface for the phytoplankton to use. And so as these phytoplankton grow, they supply food for small fish, which is in turn are food for all of those fish that make up our fisheries. And so how fast the wind is blowing, as well as its direction, can determine how many nutrients are delivered to the surface, and subsequently how productive the phytoplankton and our fisheries can be. Now, these figures from Hickey and Banis, 2003, depict typical upwelling conditions on the left, with the wind blowing southward, and then the upwelling at the coast, and that's bringing cold, salty, nutrient-rich water at the coast. And then depicted on the right are downwelling conditions when the opposite happens, when the wind blows northward and the water is then transported towards the coast, and that results in the warm, fresh, nutrient-depleted water at the coast. In the CCS, upwelling is induced by equatorward or southward winds, and downwelling is induced by poleward or northward winds. And so these winds are forced by an atmospheric circulation around the North Pacific High, whose position and strength vary seasonally. And so as a result, typically upwelling in the northern CCS occurs in the summer, so in this figure on the top left. And you can see that the downwelling is positive, upwelling is negative. And then downwelling occurs in the winter. And then in the central CCS, upwelling typically occurs year-round, with the strongest upwelling happening in the summer, as shown here in this middle left diagram. Upwelling events can occur during downwelling regime in the north because a wind reversal over a period of several days is enough to induce upwelling. And same is true for downwelling events during the upwelling regime. I mean, if you've experienced weather in general on the West Coast, it's not always blowing constantly in one direction. So keep in mind that those events can happen. And so now back to the phytoplankton. Since the phytoplankton use nutrients supplied by the coastal upwelling, you might expect that the most productive times would be when the winds are the strongest, for example, off of central California in the summer. However, previous studies have found that productivity is limited when there are extremely high winds. So for example, in this idealized mixed layer conveyor model, Bachelor et al. In 2003 found that if upwelling winds are too strong, the phytoplankton are swept off the shelf before they can utilize all of the upwelled nutrients. And this process yields a dome-shaped relationship, which is shown here on the left, between phytoplankton productivity and wind speed. And a similar Bachelor et al. Study in 2006 found that intermittent winds can prolong the time on the shelf and increase that optimal wind speed. And so to understand this, why this might be, let's imagine that we're in a sushi restaurant that has a conveyor belt that delivers little plates of food to your table, and we'll pretend that it's in non-pandemic times. And so let's say that you're sitting and waiting for something to catch your eye when you see it. Your absolute favorite type of sushi is coming down the line. You're watching it with anticipation, hoping that no one else will grab it, but the conveyor belt is moving so slowly, and it's taking forever, and your sushi is still about 4 people away, and it seems like it's been a full minute since you first spotted it. And so that's terrible. I wouldn't be happy. And so you talk to the sushi restaurant staff, and you ask them to just turn the conveyor belt speed up a bit. But now the plates are flying by. You spot your favorite sushi, but it flies by in seconds, and you barely got a finger on it. And it goes by again, you still can't grab it. And this is terrible. And you still can't get anything, and you're still very sad. So third time's the charm. You talk to the staff again, ask them to turn the speed down just a little bit. And so here's the test. You spot your favorite again. This time, the belt is moving at a perfect pace, and you're able to grab your favorite with ease and without waiting too long. And so now you're happy and you're full, and this is now your new favorite restaurant. So back to the phytoplankton, the high winds are bad for productivity because the phytoplankton respond to the wind in two independent ways, through the nutrient supply and export offshore. And so with upwelling favorable winds, which are depicted on the x-axis, with the winds increasing to the left, higher wind speeds mean more nutrients are upwelled onto the shelf. So this is depicted by this red dashed line. However, in terms of export offshore, the higher wind speeds mean more export offshore due to the Ekman transport, resulting in less phytoplankton on the shelf, and that's depicted in this blue dashed line. And so the Botsford relationship represents the phytoplankton's response to the balance between the upwelled nutrient supply and the offshore export, both of which are driven by the wind. And this balance ultimately results in a dome-shaped relationship between wind and chlorophyll, and that's plotted by the curve of chlorophyll concentration as our proxy for phytoplankton on the y-axis. And this relationship is further complicated by the regional dynamics of the northern CCS, which is unusually productive despite experiencing weaker winds than the central CCS. These regional dynamics include canyons that can enhance the upwelling strength, outflow from the Salish Sea and Columbia River, and retentive features. And so retention can be caused by geography, such as retentive areas associated with banks and capes, which are depicted in this figure on the left. And then outflow from the Salish Sea and Columbia River can provide additional nutrients for the phytoplankton, resulting in a higher productivity. So these maps from Davis et al. 2014 demonstrate the importance of this outflow, with the reality plotted on the left here, productivity without the Columbia River plume plotted in the middle, and then productivity without outflow from the Salish Sea on the right here. And you can see there's a pronounced drop-off in drop-off in productivity without the outflow of the Salish Sea. And so, using the chlorophyll concentration and wind stress derived from satellite observations, we analyzed the relationship between chlorophyll concentration and wind patterns in the CCS to see if this dome-shaped Botsford relationship applies to the CCS as a whole, and whether it applies to the northern CCS or if there's something else going on in the north. In addition, we investigated how retention affects the relationship between chlorophyll concentration and wind stress in the northern CCS using results from a particle tracking experiment conducted in a ROMS model of the northern CCS. And so first, I'll talk about the satellite analysis that we used to answer this first question. So we use chlorophyll concentration as a proxy for phytoplankton biomass. And this is derived from ocean color from a merged satellite product from the European node for global ocean color. Meridional wind stresses were calculated from QuickScat and ASCAT satellite wind velocities. The chlorophyll concentration data were 8-day composites spanning 2000 through 2019 at a resolution of 4 kilometers, while the wind stresses are 8-day means over that same time period. And this analysis focused on the shelf region from the coast to the 150 meter isobath, which is shown here in red. And to address the spatial variability, we split the region into 1 degree latitude bands, which are shown in these colored boxes. So this orange one down here in the south, and this blue one up here showing an example from the north. So for each of these, we took the mean, which resulted in 1 time series for each degree of our entire domain, 35 degrees north to 50 degrees north. Lastly, the chlorophyll concentrations were binned by the corresponding wind stress for each latitude. And so plotted here are the resulting chlorophyll concentrations for each wind stress bin after eliminating undersampled bins. So in this figure, the warmer colors are representing the central CCS and the cooler colors are representing the northern CCS. And upwelling favorable winds are the negative wind stress values southward, and downwelling favorable winds are the positive wind stress values on the right side of the plot. And so over the whole CCS, the relationship between chlorophyll concentration in wind stress is dome-shaped as suggested by Botsford. But individually, the results in any single latitude do not really support this Botsford relationship, except perhaps at 42.5 degrees north, which is that bold yellow line that'll be a little bit clearer on the next slide. And it kind of shows a hint of this dome-shaped relationship between the wind stress and the chlorophyll concentration. However, over the whole CCS, the relationship between chlorophyll concentration and wind stress has a dome-shaped envelope with individual latitudes contributing different parts of the dome. And furthermore, the central and northern regions have significantly different relationships between mean chlorophyll concentration and wind stress. This difference is evident in the separation between the mean curves for the central and northern regions, with the central plotted by the shaded red and the northern plotted in shaded blue. And this represents the 95% confidence interval for the central and northern regions. And while you can see that these curves do overlap a little bit at a few points near 0 pascals, the rest of the points are significantly different. And again, this gray curve represents at 42.5 degrees north. And this kind of splits between these two regions. And it's one of the only latitudes that experiences almost the full range of wind stress values for the CCS, at least frequently enough that we could have a fully sampled. And the difference in wind stress at which the peak chlorophyll concentration occurs suggests two dynamically distinct systems, north and south of 42.5 degrees north. The central CCS appears to be governed by traditional upwelling dynamics, as described by Botsford, while the northern CCS is a bit more unusual, with these high chlorophyll concentrations observed during times even when there's weak upwelling favorable winds or into the downwelling favorable winds. Furthermore, unlike the central CCS, the northern CCS has high chlorophyll concentrations during these positive times. Oh, I just said that. And the mean chlorophyll concentrations at these positive wind stresses, there we go, are almost as large as the ones that you see in the central CCS at more typical upwelling favorable wind stresses. And this pattern goes against the traditional upwelling driven phytoplankton productivity that is kind of characteristic of the CCS and other upwelling systems like this. And it suggests that there's another mechanism supporting the phytoplankton biomass in the northern CCS. And so why is the northern CCS productive at these positive wind stresses? Hickey and Banis in their 2008 paper present two hypotheses for why the northern CCS is so productive at these weaker wind stresses. And I kind of touched on these briefly in the introduction. So retention on the shelf that would allow the phytoplankton more time to use these upwelled nutrients could explain these patterns. The increased retention could be due to wind intermittency as described in that Botsford 2006 study, geography or mesoscale features like the retentive regions near banks and capes, and that could result in having a higher chlorophyll concentration at lower wind stresses. Another example could be the non-upwelling derived nutrient sources, such as that outflow from the Salish Sea and the Columbia River in that paper from Davis et al. In 2014. And so we're revisiting the idea of the wind having two independent effects on phytoplankton as a supplier of nutrients via upwelling and as an exporter off the shelf via that ekman transport. The retention would help counteract that the export effect of wind, while the non-upwelling derived nutrient sources would just enhance the nutrient supplying effect of the wind. And so using our ROMS model, we were able to test this first hypothesis regarding retention in the northern CCS. And so the Cascadia model is used in these particle tracking experiments, and it is an 8-year physical hindcast model spanning 2002 through 2009, and it utilizes the Regional Ocean Modeling System, or ROMS. And it was developed under the NUTOX program by the University of Washington Coastal Modeling Group. Its domain is shown here in this figure, and it's from the figure from Giddings et al. 2014, and it includes the Salish Sea and coastal oceans and estuaries of Washington, Oregon, Vancouver Island, and British Columbia, with a horizontal resolution of 1.5 kilometers at the coast and 40 bathymetrically following vertical layers. And this is kind of like a very brief overview of the model details. And there's a few papers you can look at if you want to know more, specifically Giddings et al. 2014, Davis et al. 2014, and Sidlecki et al. 2015. And so we conducted a particle release experiment in this model to test the retention hypothesis. So we had the particles released on the surface above the 150 meter isobath, so on the shelf, every 0.05 degrees of the model domain, which is shown here on the left, and those release points are plotted in black. They were released every 10 days over 2003 to 2009, resulting in about 58,536 particles per year. And so once they were released, the surface trap particles were tracked for up to 350 days. And more information about particle tracking experiments can be found in Banis et al. 2009 and Stone et al. 2018. And, there we go. And so retention times were calculated for each of the particles based on how long it stays on the shelf or above that 150 meter isobath. So in the figure, we have examples of short retention times plotted in blue of less than 5 days. And then the longer retention times are plotted in red, and those are greater than 25 days. And these times were averaged monthly for each latitude band to correspond with the satellite data. And so based on the Hickey-Banis hypothesis, we expect a complicated or dome-shaped relationship between the retention time and wind if retention is driven by intermittent winds or those geographical features as hypothesized in their paper of retention at capes and banks like that. And so in this figure on the left, retention time on the y-axis is plotted against wind, which is on the x-axis, with the latitude denoted in color. Contrary to our expectations, the relationship between retention time and wind is pretty simple, with retention times increasing with positive wind. And so this suggests that the relationship between retention and wind is dominated by the effect of onshore Ekman transport that occurs during downwelling, or that positive wind stress, and that would just keep particles on the shelf. In the figure on the right, chlorophyll is on the y-axis, and it's plotted against the retention time in the x-axis. This plot shows that the highest chlorophyll concentrations occur at the lowest retention times, with the chlorophyll decreasing with retention time. And so overall, it seems likely that on a large scale, retention is largely driven by that simple mechanism of the downwelling favorable winds inducing Ekman transport onshore, rather than this more nuanced causes of retention, such as wind intermittency, geography, and mesoscale features, as hypothesized in the Hickey-Banis paper. And while these results found that the effect of the wind intermittency on retention is minimal on this 1 degree wide latitude scale, it does not really disqualify the influence of more complex controls on retention, like wind intermittency, on smaller spatial scales. It's just on this large scale view, it's not really a factor. And so to conclude, we found that the relationship between the chlorophyll concentration and wind stress in the California current system is indeed dome-shaped, and that there are two separate curves that describe the relationship between chlorophyll concentration and wind stress in the CCS. And then that the higher chlorophyll concentration is observed in the northern CCS during these weak upwelling favorable and downwelling favorable winds. However, on this 1 degree wide latitude scale, the retention is an unlikely reason for this high chlorophyll concentration observed here. And so from here, we proposed a revised Botsford relationship. And so the central CCS follows the traditional upwelling dynamics as described by the Botsford relationship, which is shown here on the left, with the orange curve representing that relationship between chlorophyll and wind stress. However, the northern CCS behaves a little differently due to both its upwelling derived and non-upwelling derived nutrient sources. So in this case, you have these non-upwelling derived nutrients like that outflow from the Salish Sea is bumping the chlorophyll wind stress curve up a bit into those, pushing it into the downwelling favorable wind conditions. And that's depicted here on the right with this teal curve. So this mechanism could explain the unusual productivity of the northern CCS at the weak upwelling favorable and downwelling favorable winds. However, further investigation is necessary to test this hypothesis. And so to end, I just want to thank my advisors and lab groups, the Banis Lab and the UW Coastal Modeling Group, and to my funding sources over the years, the IPOC program, NSF GRFP, and the NOAA Meerkat program. And that I will take questions.
Thank you, Hally. That was wonderful. I really appreciate your presentation. So audience, we have about 5 minutes to answer your questions. We encourage you to ask them. So take the next minute or so to enter them into the questions box, which is on the right side in your control panel. And while you do that, I just want to remind you that Hally's also very kindly provided her slides as a handout. So before we close the presentation, I would encourage you to download them from the handouts menu, which is also in the control panel. So sometimes there's a little bit of a wait, and we're going to give it a second for people to ask their questions. And while we're waiting, I'll also remind people that if you know anyone who might be interested in Derek or Haley's work, please share the recording of this webinar, which I'm going to upload to the NOAA Central Library YouTube channel in just a few hours. Oops, I heard some. Oops. All right, just going to get another minute. See if we get any questions.
Yeah, I know mine wasn't as exciting about sharks like that.
No, I think that it's just both really exciting. I just think it's questions, you never know how people are going to engage. I did want to also add, you cited a number of really interesting articles, and if anyone is interested in accessing those articles, please contact NOAA Central Library, because we do have access to many of them, and if we don't subscribe to some of the journals, we can always get them through interlibrary loan. All right, so I think we're gonna go ahead and end the presentations. Did you have any last comments, either Derek, Hally, or Drew?
Just thank you for this opportunity. It was great.
I really appreciate you presenting your dissertation work. It's really fascinating, honestly. I'd like to conclude by thanking our speakers for sharing their research, and thank you, Drew, for the introductions as well. Best of luck to all of you with your Knauss fellowships. And audience, I truly appreciate you joined us today for Library Seminar. NOAA Central Library is proud to present the work of the NOAA community and to support the Knauss Fellows. We hope you'll join us for the next Knauss Lunch and Learn webinar on Thursday, May 20th at noon Eastern Standard Time. So until then, be well, and thank you for joining us today.